base priming to be used. Sige. You should emphasize the 3' and 5'. Okay, that we said we always consider the anti-parallel orientation of the strand that is from 5' and the other one is 3'. Pwede nyo rin i-discuss yung nucleotide that it consists of the bases on the central axis with its complementarity, meaning if this is thymine, din dapat kabisado nyo yung color na, and guanine is to cytosine. Okay? And perpendicular to this are the sugar and the phosphate group. Okay. So, how will you say that replication will occur? Paki-angat nga muna. Huwag yun ang video, intindihin nyo na lang. Ayan, tanggal na. Ano ba yan? Eh, mali naman yung nilagay ninyo, hindi yung graph. Kaya nalalaglag.
Naku, ano? Mali na venture natin. Fail.
Oo nga.
Smooth lahat. Fail.
Pahiram nga nung ano, para hindi nalalaglag. Yung kabilang group, pahiram ng 5' ninyo. Palitan nyo to.
Dapat yung ano,
Yung graph, kasi
yung graph.
GG.
5' to, no? Isa pa, 3', isa pa.
Dapat yung ano, rough.
Ang ganito.
Ito yung
Magaspang. Hindi.
Paki-angat.
Smooth yun.
Ito yung old strand, ito yung magiging new strand. Lalagay sa ilalim. Para lang kayo naglalaro. Sige. Okay. So you can mention the 9mer that there must be the presence of the repetitive sequence of bases which consists of 9, so meaning 9mers, so that the DNA A will bind to the 9mer, of course with the ATP hydrolysis. So there is what?
Smooth po. Yung borrow na lang po sa ibang.
Huwag na, mamaya na. Kunyari lang, bayaan mo na siguro. So it will cut or nix the sugar phosphate bond and it weakens the hydrogen bond, forming the replication bubble, and extend its unzipping and form the replication fork. And of course you need what? Hindi pa. Anong function ng SSBP? It requires the SSBP to stabilize the open conformation, and of course due to the unzipping there is an uncoiling ahead of the strand, so you need the DNA gyrase to relax the supercoiling. Ilalagay nyo isa-isa. Sige. O, anong susunod? Ay, hindi na natin igagawin kasi replication lang eh. There should be the complementarity between the old and the new strand.
Kung kada po isang ganun, kailangan po may DNA, pang stop.
Hindi, mention nyo na lang. Kasi you just mention it once. Meaning, every time unzipping, may supercoiling, merong magre-relax na gyrase. Every time you open up, meron siyang helicases, DNA A, DNC, unwinding protein. Automatic na siya, but you can just mention once. Pwede na yun. O, ano susunod? After the 3'. So, what is the direction of replication? Direction.
5' to 3'.
Okay, 5' to 3'. Naku, talagang bautista, ikaw mauna.
Yes!
Ay, naku. Sa mama.
Okay, so if this is the 3', now there is an anti-parallel orientation of this strand. The new strand should be 5'. Okay, so the direction is from 5' to 3'. Okay. So since there's an exposed 3' and the function of the DNA polymerase 3 is to add nucleotide. Ito yun, di ba? But then it cannot bind to the 3' end because of the absence of free 3' hydroxyl end. So it cannot bind. It requires a primer catalyzed by the RNA primase so that the DNA polymerase chain can add nucleotides. So put the complementary base. So if this is adenine, oh, ito.
Ay, adenine po.
Guanine daw. I mean, Oh, then thymine is to adenine, and then teka muna, isang unsip lang siya.
Wait.
Anong process to?
Okay, mahaba talaga. Okay. Simultaneously on the other strand, replication should also occur. And since again this is a 3' end and there's an absence of free 3' hydroxyl end, it requires again the primer catalyzed by the RNA primase. Asan na yung primase natin? So that the DNA polymerase 3 can bind to it. Asan na yung polymerase 3? Okay. So you add nucleotide, put the complementary base. Ito.
Sa kabila.
Okay, so thymine is to adenine, guanine is to cytosine. Okay. So unzipping continues. Kaya ayusin ninyo. Okay. So no expose, no expose the prime end, so there is a continuous synthesis of the DNA. So you just add nucleotide if this is guanine. O, lagay. Kunyari, nagkamali. Sige, pagpalagay na natin. Put the complementary base.
Ay, wala na pala dito.
Okay. And on the other strand,
Tulito ako sa kulay.
dapat kabisaduhin nyo yung kulay. On the other strand, saan may primer? Wala nga. May primer dyan, ha? So again, what?
Yung yellow.
Pampalipon.
Teka muna, di ka pa pwede mag-add. This is an exposed 3' end. Therefore,
Yung
it requires again another primer
Primer.
because the DNA polymerase cannot bind to it.
Sorry.
RNA, the primer is catalyzed by the RNA primase, and the DNA polymerase 3 can add nucleotide. Add. Ito.
Ay, dito na ba? Grabe.
Okay. Third and zipping. Intindihan nyo rin yan pag nag-practice na. So, continuous synthesis of the nucleotide, and we call this as the leading strand.
Kaya po ni Steph yan.
Okay.
Ikaw nga una eh.
Tama.
Pagbibigyan ka na nila, una ka na.
Tama. Sinuwi po yun.
Sige, pagbibigyan ko na kayo ladies first.
And then, it requires again the primer,
Primer.
catalyzed by
By
the
the RNA primase.
so that the DNA polymerase 3 can add nucleotide. Sige, bigyan mo.
Paano saan yung DNA polymerase 3?
Mamaya ka
Pang-add
na
ng nucleotide.
Mamaya ka na
Pang-add?
Eh, ba't mo tinanggal yung
Hindi, pwede mo
primer?
yung pang-add.
Inilog ko lang, ma'am. Para
Eh,
mas
mali
yung
kasi
primer?
magtatanggal tayo ng
Ay,
primer,
may-
huwag ka magtatanggal.
nakapatong po kasi yung tinuro niyo dito eh.
Sige, primer.
Sige
Oh,
mo
yun
no.
lang.
Yung... Grabe, daming kulay.
Okay. Sige.
Daming kulay, man.
So,
Bad color.
proofreading. So if ever there are mismatch in between the bases, now proofreading should be under the direction of DNA polymerase 1. Now DNA polymerase 1 can will act on the erroneous mismatch between the bases in the 3' to 5' direction. So titignan ninyo isa-isa. Ito. So if this is guanine, so it will be pink, and this will be replaced by cytosine. It fills also the gap. So meron pa ba? If this is guanine, it's supposed to be cytosine.
Cytosine.
Okay. So tignan natin kung may mismatch. Ito yan eh. So pink pa rin ito.
Ang volunteer na po si Amery. Muna.
Oh, sige. And at the same time, there is a removal of the primer. So the DNA polymerase 1 will remove the primer and fills the gap if this is cytosine. So this will be complemented by guanine. And if this is cytosine, it should be complemented by guanine. Okay, remove. So this is thymine, it should be adenine. If this is thymine, mali, ayan. na lang. Okay, so replication is completed. And by the way, this is sealed by the DNA ligase. Asan na? Oh, ligase. Okay. And then replication is completed, which results in one old, one new strand, one old, and one new strand. And the mode of replication we use is semi-conservative.
Ayaw pala semi-conservative.
Pero nakakagusto mo agad.
Hiramin nyo yung, eto, mag-practice kayo.